Hello.
Hey, LJ. Hi, this is Amy.
Amy.
Yeah.
Yeah, how are you doing?
Good. Are you able to see the slides? I saw you use the phone to call in
No,
like
I'm
me.
I'm going to sign on the Zoom right now.
Okay.
Shouldn't be too much problem.
And you can see we have your big profile picture on the first page already.
You're kidding. I mean, that's the good picture, right?
Yes. We got the
No,
best one.
this picture is not very good, Sammy. This is not very good.
What do you want? Which one you want?
Oh, the one, if you look at this, my presentation, it's much nicer. This looks very sad. You know, somehow it's not a very happy person.
This is the happy version.
Sorry?
I think this is the happy version, LJ.
No, if you look at my let me show you. I'm going to share. You're going to give me the screen later on, right?
Yes, that is correct.
Okay. So you're going to go through this first, the slides.
This is like the actual opening slides as we chatted, right?
Yeah, but I noticed you have six slides here. Are you going to go through the 6 before you give it to me?
Yes. Well, be very quick, like introductory. And number 6 actually is repetitive. Jerry,
Yeah, I'm just placing a placeholder.
Oh, I see what you mean. Okay. Okay.
Good. Okay. Do you want me try just to share, then you can get back to you, see if it works or not?
Yes, we should.
Okay. Let me see. Oops. Host disabled participant sharing. You have to give it to me. You have to give it.
Can you make him a co-host?
Yes, I'm trying to make co-host, but LJ, what is your account?
Which one
sorry,
is
I cannot hear you.
I just saw it. Okay, make co-host. How about, could you try that?
Okay. This will stop other screen sharing. Do you want to continue? Yes, right?
Yeah, go ahead.
Okay, share. All right, let me show you what I have here. Can you see it?
Mm-hmm. We can see your slides now.
Look at this picture. Look at this picture.
Do you want us to change it? I think both are nice. Okay.
No, that's okay. Anyway, so let me go back. So can you actually go back to yours?
Can you do a presentation mode just to make sure?
Oh, yeah, I will do that. Let's see. To design slideshow. Yeah, here it is, right?
Perfect.
Okay, I stopped sharing now, right? I stopped sharing, right?
Mm-hmm. And he can change back. Yeah, it's very easy. Yeah.
Okay, thank you. That's wonderful, wonderful.
Good. I think we should be good. The audio is good. The slides are able to change back and forth. Let's make sure.
Let's give it a couple more minutes until like 12 when more people join. People are probably signing off their meetings right now.
Oh, I saw a lot of people joining already. I mean, we as a host and presenter, we usually join earlier to make sure everything runs smoothly logistically. I'm glad to see audiences. You guys started joining beforehand too, so that's good.
Yes, hi, this is Bill. I'm new to the group. Bill Scully, I'm with Glenn Choice.
Oh, hi, Bill. Thanks for joining us.
Yeah, thanks a lot for inviting me to join the group.
Yeah, we actually have a very active membership, and every month or so have a different topic. So please make sure you signed up with the email blast. So every time you don't need to be forwarded, and then the email blast will give you updates.
I did.
Yeah. The same for all the other new members who joined. And in today's meeting, I will also, besides introduce today's session, I will also give a heads up on next month's KOL sessions or the online sessions.
Yeah, but Bill, this is LJ. I thought you need saving at least $1 million to be able to join this club. Is that right?
It's in the mail, and I wrote it out for 2.
Yeah, we have a referral. We just lower our fee to like 5,000. I'm kidding.
Oh, only $5,000? Someone told me I need to be super rich or relatively super rich, by the way.
I sent
of course,
it to
$1 million is
You sent it to your
Sorry?
Cambridge account.
Yeah.
I see.
This is a billionaire club that we're joining.
You guys are going to scare all my audiences away.
Don't worry, we're going to clarify that later.
LG, you're always so funny. Yeah.
I'll just keep everybody muted because we're having more people joining. Sammy, you feel free to unmute yourself. But LJ, I think you're calling from, your audio calls from your phones. I wasn't able to mute it, but I think this fine because you're.
Don't mute my number. 617. That number is mine.
Yeah, I got that. Somehow I can't.
Yeah,
I can't mute like phone calls. But anyways.
Oh, good. You know, because I didn't vote for Trump this time, so government is nice to me now. So they don't allow you to do anything on me.
That's good for you.
I'm not so sure. I'm worried about my income tax will increase dramatically.
You know, we used to host sessions where we have lawyers. We can help you connect.
Oh yeah, that'd be good. Yeah, my son is a lawyer anyway, so I can have an in-house consultation. But,
oh, that's nice.
But he actually charged me much more than commercial lawyers. So.
Why did you go to your son then? I thought it's supposed to be free.
Oh, you kidding me? Oh, you must be kidding me, Sammy. That's the way of treating children is completely different. You're treating your parents, right? I mean, this is a reality. So don't expect too much from your kids, Sammy. That's my advice.
Oh my gosh, I was hoping to get more from them, rather than vice versa. But we actually in March we'll have a lawyer session related to IP and big data. So just let all the audiences know we're joking around the lawyer legal things, but in Dashu, we do have legal sessions related to data analytics or your IP. So stay tuned for our email blast for those advertisements. I think we should pretty much get started. Usually I like to start on time to make sure. Hello? So, Jare, can you mute everyone?
Yeah, I just feel like if I mute everyone, I'm gonna mute LJ too, but I'm just gonna mute everybody.
Just mute everyone and unmute him.
Somehow he doesn't have a mute button. LJ, can you speak up still? Okay, I think everybody's muted.
I can speak up. I unmute myself. Can LJ still speak? Just to make sure.
Can you hear me?
Yes.
Yeah, okay, good. I think the co-hosts are good. Okay, maybe let's get started then. Thank you everyone for joining Dashu's first session this year. We have this online sessions going on for years now, and this is our kickoff for this year at January. And I'm very happy and actually proud of myself, invited to LJ to join us. We know each other for years now, and usually LJ actually very hard to be invited for the company consultation because he is so popular and so insightful for the company discussions on the clinical trials. But he is also very, very supportive for nonprofit organization like us and give us the free KOL sessions. So thank you so much, LJ. I don't think I need to go through all the LJ achievements. This listed here is our volunteer help us put something here. But actually, if you want to know more about LJ, just Google search him on Wikipedia. He is actually one of the most important statisticians I know of in the clinical trial research. So if you want to know more his achievements, go ahead. It's all on Wikipedia. And one of the things, actually, LJ, you have so many awards you've received. I think for our fellow statisticians or analysts in this background, we all hope some of them could belong to us in the future for the younger generations. So one thing I think I was really proud to see in 2009, I remember you had the Weeks Memorial Award for statistical methods used in clinical trials. I think that was one of the most prestigious award among all the international statistical societies. So you definitely are one of the role model for all of us in this area. So thank you for accepting my invitation to join us. So today, LJ will talk about one very important urgent issue about COVID-19 disease in clinical trial studies. So I'm not going to go through all the detail for that. LJ will definitely give you guys the updates. But maybe before we move to the presentation with LJ, I do want to introduce Dashu a bit more in case the new members didn't know and how to contribute or be part of the Dashu Society. So as I already mentioned, we are a nonprofit organization. We also have strategic partner in different organizations, co-host or hosting our own events. We truly really wanted is to increase the influence of analytic society, including statisticians, data scientists, or biostatisticians, all of them. So we have different type of topics or even career development events every year. And monthly, we host this online online events for free, and membership is also for free. Most of us actually come from either industry or academics as leaders in different area, try to support this event. Myself, most people maybe know me from different sessions already. My name is Sammy Tang. I heads up a department of statisticians and biometrics in a pharmaceutical company called Sevilla US. And we also have different volunteers supporting us from students' societies. So please feel free to join us. Either you want to be a volunteer or you want to just listening to some of the sessions. You can see we have recorded sessions when agreed by the presenters on the Dashu YouTube channel, and also you can email us to contact us to be part of the member, which is totally free membership. We do need your support, as I mentioned. So if you want to be a speaker, if you think your presentation will be very helpful, contributor to the society, feel free to reach out to me or any of the people you see on the email. So, Jerry, can you mute this person again? I think a new person joined. They were not muted. But anyway, you can see all the volunteers. We actually have happy events together, and for students or for the committee members and for the board members. And I think the volunteer put my picture very big over there, and that's myself. So it's a happy community. Please join us and let us know if you have suggestions. We'd like to grow together. And our final goal is just to contribute support back to our community as a whole. Next page. Just give a quick heads up. Every month we have event. I also want to advertise a bit of February event Jerry will be organizing. We have Frank, Dr. Frank, to present the genomics from the Ohio, to present the machine learning based interactive multi-omic analysis and for NGS data processing. Actually, he is also very active in the community and the co-chair for CAPS. So with that being said, I don't want to hold on this too much. Really want to give the floor to our star today, LJ. So the floor is all yours.
Thank you very much, Sammy. Let me, let's see, share the screen. Can you see my screen? Yes?
Yes.
Okay, good. Okay. Well, I really appreciate this, LJ Wei. Sammy, very generous inviting me. I know a lot of people try to give talks, but I think they have a beating war or something, but my friend Lu Tian, actually at Stanford, he probably is vice president of this organization. So actually he probably forced Sammy say, listen, you need to invite my old friend LJ to give a talk. So I appreciate it very much. Today Boston is very cold. This reminds me when I was a student many years ago in Wisconsin, and pretty windy too. So time goes so fast. I came to this country in 1971. You can count how old I am right now. Just amazingly, we can see so many young people join our profession and doing so well. That's really a pleasant feeling as a older. Forgive me to say this. Sammy said I'm a Harvard 元老. I think this is 元老. I apologize to my American friends. 元老, it sounds like old guys. Probably should hiding in the back now. Maybe we should thinking about some nicer alternative. In any event, I will discuss with you today is very interesting. I have to confess, about maybe less than a year ago, I didn't know much about COVID-19 stuff, neither about clinical trial for COVID-19. COVID-19 trial is very unique, so I'm going to share with you. But about maybe 2 or 3 months later, we actually gradually understood a little bit. And so we wrote the papers together with Lieutenant or my other young colleagues and submitted to medical journals. It turns out everybody's were so hungry about getting publications COVID-19, so we can easily squeeze in. So I'm going to share with you what we learned in the past less than a year. Towards the end, I really wanted to share with you some very exciting area in COVID-19 trials, especially for outpatients. The outpatient trial is quite different from inpatients, and we can talk about this towards the end. So I try to finish within 40 minutes, so I'll give you time to ask questions or we discuss some issues. So let me just share with you, this is just three of us, but honestly, more people get involved in our research group. Zach is a very young man. It's just a graduate from our department a couple of years ago. But this guy just, like most of you guys, he doesn't sleep. And he actually published probably 30 or 40 papers in the past a year and a half in the top medical journals for statistical publication, not for clinical publications. That's amazing. Of course, my old friend, Lu Tian, now he's a Stanford professor. He's a genius. Everybody knows that. Every day, almost call him up. I hope he didn't get tired of me, and I just want to squeeze his brain a little bit. So after I talk to him, I can talk to other people. So I'm just like a middleman, just make a few bucks in the middle. And let me, I was told from the Zoom, there's a connection. I better show up my face. Unfortunately, my desktop doesn't have camera. So I still try to figure out how to do that. Especially, I'm started teaching this spring semester, and a student complains that I want to see your face. So let me just show you my face. Sorry. So this is a nicer picture than the one Sammy posted. The picture was taken several years ago in Japan. The reason I put up this picture, I went to this barber shop in Japan, in Tokyo, right? I couldn't speak Japanese, so the barber didn't speak English, or neither Chinese. So I said, I'm in trouble. I don't want this guy give me a haircut. Turns out this didn't look good, right? So I called my old buddy Hajime Uno. He was in Tokyo that time. He's Japanese, so he came over as an interpreter for me. And the poor guy, the barber, spent about an hour and a half cutting my hair piece by piece and used scissors. He didn't use a razor at all. So afterwards, I said, "Oh my goodness, look at this hair! If you can compare the picture I had, that was done by an American barber. It was really rough, but look at this one, just beautiful, right?" So I want to show you. Sometimes I look good. Okay. Now, I have a recent hire assistant, and she's an energetic little lady, and 2 years old, and just happened to be my granddaughter, and 2 years old.
Oh, so cute.
So she actually helped me quite a bit. Is a lovely lady, just about a mile from us. Unfortunately, because of the pandemic, we could hardly see her these days. All right, let me start with one slide talking about this is a word called estimate, right? Then in general, then we're going to go into the COVID-19 clinical trials. So what is estimate? Estimate, probably everybody knows right now. It's very confusing. Sometimes we don't even understand what the ICH talking about, right? So ICH E9, the R1, actually they put some extra statistical guidance, right, or some kind of sensitivity analysis. Particularly, they actually introduced this idea for estimate. Estimate actually composed for 5 components. Let me just pick up 4 of those guys. Of course, every study you needed to specify population, right, patient population. Then, of course, then you have to define the study endpoint. And the next one is after study endpoint, you have to define how to summarize those treatment difference, right? Hopefully those study endpoint study summary have some kind of clinically meaningful interpretation. Otherwise, we're all in trouble. So today I'm going to give you examples. Even for COVID-19, people use some kind of a quantity we actually very difficult to translate. And the last one, of course, how to analyze the data. So ideally, you really want to have a robust, efficient way to analyze our data. But it's easy to say those kind of things. But I tell you, you guys are really experts in the field to actually design those, even those components of estimate. It's a very hard task, right? Everybody knows that. So I'm going to say this a few words. COVID-19 trials, COVID-19 actually helping the clinical trialists to really understand, to start to wondering what we have done in the past may not be ideal, right? This guy, Don Barry, Professor Don Barry in MD Anderson said a couple times in public. He said, listen, guys, the statistical methodology research in clinical trial area actually is led by two groups of people. One is industry people. The other one is regulatory people. Academic people, they don't do much clinical trial methodology research anymore. So I was so disappointed to listen to him. Afterwards, I talked to Barry, Professor Barry. I said, wow, gee, Don, why you want to say that? He said, well, give me an example. Who actually doing clinical trial in the university now? But he's right? He absolutely right. So the reason the clinical trial methodology, we actually don't do much in the academic world because NIH funding, they concentrate a lot about high-dimensional protein or genetics research. That's a very fancy, right, sexy area. And they thought a clinical trial, just like routinely, right? You just push a button, then everything's fine, right? Turns out in the past 5 or 6 years, our group found out a lot of interesting stuff. We haven't actually be really exploring, can we improve our practice or not, right? So today I'm going to use this COVID-19 situation to share with you what we really learned from COVID-19. Now, first, I'm going to just talking about several trials. They actually finished. It's a very high-profile trials, and also I'm going to talk about some trial actually ongoing. And then towards the end, we're going to bring in this outpatient situation we're talking about. But first, we're going to talk about in-hospital patients trial. So now, those trials is all relatively very short. It's 28 days or 30 days, right? Because basically, if you don't recover like 28 days for months, we are in big trouble, right, with the flu. For mortality information, probably people just extended to 60 days or 90 days follow-up. And this is actually very interesting. This whole clinical trial in COVID-19 belongs to so-called critical care medicine. Honestly, I haven't done anything like before 2019 in anything about critical care medicine. Everything we did is a long-term follow-up, right? Cardiovascular trials, the cancer trial, anything you name it. Nobody actually give you like 20 or 30 days, except for this very short phase 2 trial for oncology trial for response rate stuff, right? So this is a fascinating world, very short, and we like to find out how do you define the endpoints even, right, for this short-term trial. Now, first, let's see how to select an endpoint. Now, I'm going to give you a couple examples. They actually have a binary endpoint. For example, on the day 14 after randomization or day 28, you define an event. You just say, okay, I'm going to use this binary, either the guy had an event or not, right? They also use a continuous variable as an endpoint. For example, the duration of a clinical event. For example, duration of hospitalization, right? Duration of ICU staying. Also, you use a time to event. That's very interesting. So why in the clinical trial in COVID-19 have talking about time to event? And also some people actually really interesting to know, especially when you're dealing with outpatient, the interesting viral load actually reduction. Those are biomarker, of course, is debatable, right? How did you translate the viral load reduction into clinical outcome? Now, the third one is a very specialized in COVID-19. COVID-19 is very interesting. people actually, the WHO, World Health Organization, they actually created so-called ordinal categorical outcomes from the clinical stage. Just let me give you one example, just a few minutes, right? This is very unique for this older clinical trials we face. Now, after you define the endpoint, either binary, continuous, or ordinal categorical outcomes, the first thing is say, for example, in the treating arm only, right, separately from a placebo, can you give me a reasonable summary of this so-called distribution of your endpoint? Of course, everybody says, oh, yeah, I can use a mean, I can use a median, I can use the event rate, right, like 14 or 28 days. Or some people actually can actually call the hazard, right, like a mortality. And also, you can talk about intensity rate instead of a hazard. So you can summarize each group by a single summary. But unfortunately, in the ordinal categorical outcome, like we said in the last one, this one is very difficult, right? We're going to talk about this is actually clinically very meaningful endpoint. Unfortunately, statistical speaking, there's also some kind of issues we have very challenging. So now, after each group you summarize by means, by median, and etc., then you say, how do you summarize the between group difference, right? Between the placebo and your treating arm? We say, well, let's talk about a mean difference or mean ratio, right? This endpoint. You can talk about median difference and etc. Event rate difference, right? And people use a hazard ratio even, right? Or event ratio and etc. So now we actually have all kinds of endpoints. We have all kinds of summary statistics. Now you notice those endpoints or the summary statistics, they all model free, right? Actually, we don't do any modeling at all, except for this hazard ratio. Probably we need some kind of assumption like constant hazard over time. Now let me give you, this is a typical ordinal categorical outcome for COVID-19 trials. Almost every clinical trial in hospital, they actually consider this outcome either major outcome or major endpoint, or actually is a key secondary endpoint. Okay, this is one of the choices. They have a different classification. Sometimes people use 8, some people use 6 outcomes, right? 6 categories. Now let's go through this very quickly. The first one is disease, right? So that's pretty bad. The second one is the patient hospitalized, require invasive mechanical ventilation. That's pretty bad too. The third outcome is hospitalization, require high flow oxygen. And then the fourth one is low flow oxygen. The fifth one is hospitalization, not require oxygen, but we need attentive care. That means I still need the hospital people helping me out, right? Then hospitalization, the sixth one is not require attentive care. That's actually getting better now. That means this guy is on the way to recovery. Now the last one, of course, is the guy is discharged alive, right? So you notice the lower category, the worst, right? Some people actually the other way around, right? Put the best in the top, right? Whichever you want. So this case, we're going to use this one as an illustration from the worst to the best, okay? Now, people use this ordinal outcome, did a trial. I don't know my friend from Gilead on the call or not. This is one of the very high-profile trial sponsored by Gilead. Of course, everybody knows now that they have this famous the drug now, remdesivir. This study was published in New England Journal of Medicine about the last year, right? So what are the treatments? The 5 days treatment against 10 days treatment. Okay, that's the duration of the treatment for two arms. What is the primary endpoint? The primary endpoint, they use an ordinal categorical outcome on day 14. So what did they do? Actually, they did this way, just 1 to 7, right? And you say, well, this is my the major endpoint, and I wanted to know what kind of distribution for placebo arm and what kind of distribution for treating arm. So let me provide you the data for you. This is from the paper anyway. So let's see. This is 5 days. The first column here, 200 patients involved. 10 days, 197 patients involved. Say 1 to 7, right? Category. First row is the number of people died, 16 against 21, 8% against 11%. Okay, 5 days looks slightly better than 10 days numerically, right? Then you go down and go down. You look at this, the 7, right? The 7 categories. Those guys checking out alive is 60%, and against 52. Boy, this guy is also 5 days is also very good, right? Compared with the 10 days, right? So when they designed the trial, it's very interesting. When they designed the trial, they actually assuming this ordinal outcome. Then they said, well, this is a very nice, but how did you summarize the difference between the two distribution of this outcome, categorical outcome? So first, you hated to assign numerical value, say, oh, the death I gave you 1 point, and the hospitalization, this second one I give you 2 points, right? Everybody will jumping up and down, say, oh no, you cannot do in 1, 2, 3 to 7. That's very artificial, right? So people say, well, gee, what am I going to do with this? I said, well, can you consider so-called ordinal logistic regression? I said, well, what is that, right? That's actually, let me just share with you. What do they mean? They actually talking about extending this binary data with odds ratio, and turns out to be handling this multiple outcome. So I'm going to show you this result later on. The trouble is for ordinal categorical data, it's very hard to summarize each individual group endpoint profile. For example, if I ask you, look at this 5 days distribution, right? Can you actually give me a single summary, sort of measure of the center? I call this word, right? Center of the distribution. I said, well, can I use a mean? I said, I don't have a mean at all because I don't know numerical value for those guys. Can I use a median? I said, well, gee, it's too discrete, right? The median probably sitting somewhere here. So nothing is actually we can use as a single and non-parametric way, model-free way to summarize each group, right? So that's one problem we're having for ordinal categorical outcome. Now, second one, if we're assuming a numerical value for each category is too artificial, right? So the next one is how to summarize the treatment difference between the two columns, right? Ordinal categorical data. So this is the data they published. So we have 7 categories, but we're actually doing this so-called proportional odds model, ordinal proportional odds model in the following sense. We actually have a 1 to 7. I collected those older patients, their category is less equal to 1. That means they are dead, right? It means 1 is dead, right? So this is toward the end of the day, right? 21 against 16, and then you collapse all the people, their category is more than 1, right? So you have a 176 people and a 184 in those groups. Then you calculate odds ratio, which is a 0.73. Okay. Then I said, well, wait a minute, let me cut in by number of category 2. So I collapse the 1 and the 2 together. You have 54 and 32, and a grade then those you have another odds ratio, right? Calculate. Then you do the same thing consecutively. You got a 1, 2, you got a 1, 2, 3, 4, 5, 6 guys, right? So you're saying, well, I have those 6 odds ratio. What am I going to do about the totality of the evidence across all the categories? I said, well, it's very easy, just combining those guys, right, in some fashion, in a reasonable fashion. I said, okay, let's just combine this. You can use even like a mental health type of things, or using one of the variants combining. But one big assumption is, do you really think those numbers, odds ratio, they are roughly constant across all the chopping, right? If in this case, if odds ratio less than 1, that means 5 days is better. Okay, so it's a funny situation, right? Everybody thought that 10 days would be better. They actually designed that way, but turns out 5 days turned out to be better. But we don't know how to summarize those guys. So they actually use a proportional odds, the common odds ratio, as a quantifier between the two arms. That's what they designed the trial. Okay, so let's see what happened. Now, first, even the model is plausible. That means the odds ratio, the 6 odds ratios, underlying odds ratio are pretty much constant. We still have a difficulty to interpret and say, hey, geez, your odds ratio, you need a background probability, right? Event probability. I said, what do you mean by that? Because every chopping, you have a different probability. So it's very hard for people to understand the odds ratio of 0.56. By the way, most of the people, including me, I don't know how to interpret odds ratio. Usually odds ratio, if you're a gambler, you know the odds ratio very well. The odds, for example, right? But our education, we don't know much about it. You can use a risk ratio if you wanted to, right? Now, since the proportion odds assumption was not met, so they in the study, in the paper, they saying, well, after we collect the data, we find out those odds ratio, the 6 odds ratio, actually they are not a constant underlying odds ratio. So what I'm going to do, the guy said, well, the only thing, I tell you what, let's just be using Wilcoxon test, right, based on this ordinal categorical outcome. Because the Wilcoxon test, you don't need a numerical value, right, for each category. You basically just do, well, category 1 is less than category 2, and et cetera, right? So it turns out the p-value is 0.14. There's no difference. This is stratified version, by the way, okay, the Wilcoxon test. So that's what they're reporting in the paper. You say, well, guys, I only can do the testing because the quantifier, the estimate, we assumed is not valid, right? So they leave everybody's scratching their head and say, well, you give me this 0.14 in favor of 5 days, but how much better this 5 days, right? Now, everybody knows in this audience, if you have an ordinal categorical outcome, you can use this probability difference, right, to sort of like a size to measure how good you are, more than just a p-value. For example, you could say probability 5 days better than 10 days, right, based on this ordinal categorical outcome, minus the probability the other way around. If this guy is, in a positive, this fellow is 5 days is better, right? On the other hand, if you can argue, say, well, you give me this probability 5 days greater than 10 days. In fact, I don't know how big the difference is, right? Could be it's so small difference, but you still have a highly significant probability difference, right? So this is not ideal, the size, the measure of the size of the difference. So now, what is the lesson we learned from this trial? Very nice trial. And we finally realized in the beginning, when you design the trial, the ordinal categorical outcome is a good choice because clinically speaking, people use this in their brain, right, to identify you guys are doing well, you guys are not doing well, and et cetera, right? But on the other hand, if you quantify the difference between the two outcomes, two treatments, you use a proportional odds model. Just suppose a proportional odds model is not valid towards end, we're stuck, right? We don't know how to do it. So the lesson we learned is, say, can we actually use a model-free summary of the treatment difference? And that's what one thing we really like to know in this ordinal categorical. So there's a lot of interesting research still. How do we quantify this ordinal categorical outcome? Now, let me share with you other trials. The endpoints actually derived from ordinal categorical outcome, right? There's a 7 scale. This is a famous trial, and this is actually trial. This is a trial to make the FDA approve this Gilead treatment formally, and also accelerate approval first, right? So what the trial was done by NIH actually with the taxpayers' money. We are grateful. They actually used this remdesivir again. This up to 10 days, by the way, okay? And against the placebo. We call ACTT1. This is the first trial. This paper was published by New England Journal of Medicine last year in May. Okay. Now listen, this is quite interesting. Now, I believe that they actually use the ordinal categorical outcome first to design the trial. Somehow in the middle of the trial, they change it. They change into the time to recovery improvement based on ordinal outcomes. Let me go back to this one for you, okay? Now, this is very interesting. In the ACTT1 trial, they said, well, patient probably entered the trial like in the 3, 4, 5, whatever it is, right? It's 28 days, right? Or 14 days. We like to know when this patient from the third category move up to 6 or 7, right? That means the 6, 7, they call it recovery, that recovery stage. But they are pretty sick in the beginning. So that's what they are doing. They're using time to recovery. So this is an interesting idea. Let me just give you a typical pattern of those patients, right? This is the first patient had on the trial 7 days, and the green circle means recovered. So this is a patient very lucky. recovered after 7 days. And the second patient, red one, with a cross red means the patient unfortunately died on the 14 days, okay? Then the third patient is censored at 21 days. And the fourth patient towards the end of 28 days, this guy is still not a recovered. We call this also censored, right? Now, this is very interesting. Remember, they're actually the main endpoints, time to recovery, right? So here's the issue now. For the first one, I know exactly the 7 days recovery time, right? But for the second patient, I'm in trouble. The patient died, unfortunately, during the trial. I say, listen, guys, what is the time to recovery for this guy? We said, well, I'm sorry, the guy is in heaven. How do you define the time to recovery? I said, I don't know. So the ACTT1 people saying, well, in that case, why don't we give you 29 days, right? Because more than 28 days, I don't even care. You can give you 99 days or 100 days, whatever you want. So this is actually very interesting phenomenon. The time to recovery is become an issue by the competing risk from the death, right? Competing risk means the death occurring before you recover, okay? Now, the third one, of course, this is sensor observation. Now, let me just emphasize the right corner, right column here. This is what we're going to talk about later. Now, instead of we call the time to recovery, instead of we call time spent post-recovery days. For example, 28 days, this is the first patient recovered 7 days. So this guy enjoying the life for 21 days at least, right, for the rest of the study. He said, wow, great. The larger, the better, right? The second patient died before have recovery. I said, how many days did the guy enjoying the after post-recovery? That's 0 day, right? And then the third one is less than 7 days because this guy censored. And the last one also gives the 0, right, 0 days. So in this case, because 28 days follow-up, we have 2 zeros, right? So I'd like to emphasize, we'll come back here. This actually becomes a really interesting endpoint to deal with, okay? Now, here's the issue, right, we're talking about already, right? Patient died before recovery, we don't know how to define it, okay? Time recovery is not well defined for those patients. Recovery rate at 28 days may be okay. That's just the multinomial rates, right? And this is the last bullet is very interesting. We probably ask ourselves, why do you think a time to recovery is important? How come we just say 28 days recovery rate? That really matters. Actually, it's not. If I can shorten your hospitalization, right, staying, I can release the medical those resources to other patients, right? Especially when people getting really overwhelmed in hospitalization, people, right? So we do appreciate this endpoint. That means in general, if you have a shorter duration of misery, the better, right? Okay. So here it is. We actually started to plotting this proportion of recovery, the vertical scale, right? The percentage of people from 0 to 1, and a number of days on the horizontal line. And we actually plotting those the proportion of people recovered, right? But this is actually because competing risk. We only talk about those guys survived before we observed the recovery time before the guy passed away. In this case, if a patient recovered, most of the patient, every patient survived. So we only worry about the survivors, right? The time to recovery. So this is actually the incidence curve. We call this accumulated incidence curve with competing risk. Did you notice there's a bigger chunk of a probability on the top? We cannot reach it because those people died, either died or 28 days is still having trouble, right? Now, if you look at this accumulated incidence rate curve, right, the higher the curve, the worse, right? I'm sorry, the higher, the better, right? Because time to the recovery is sooner, right? So this is opposite for the bad event. This is actually time to a good event, which is a recovery. So you have the blue curve is higher than red curve. That means you can graphically, you're saying remdesivir indeed will actually shortening the recovery time, okay? So what is the trial they analyze the data? They said, well, gee, you do have this competing risk, but they didn't use a competing risk as a word in the paper, in the study. They basically, if the guy died, in this plotting, they just assigned the 29 days. That's essentially just using survivor, the same thing, right? But then you said, well, how to summarize? You can summarize the 28 days event rate, right? Or you can use the time to event analysis. Then the guy said, well, how did you summarize the time to event analysis between two curves? So everybody said, well, let's use the hazard ratio, right? But hazard ratio, this word is for bad event, right? It's not for good event. Then they changing the hazard to event rate, right? Event rate is constant. So they assuming the event rate is constant, and they give you hazard ratio equivalent. Okay, that's what their summary. That's define the difference between the two groups. So let's talk about this a bit. Why this is not a very attractive. Okay. Now this is what happened. The hazard ratio is 1.32. Remember, the higher the curve, the better, right? That's why the hazard ratio event rate is higher than 1. That means it's good, right? The remdesivir is good. So they said, well, it's a 32% the fasting, right? The faster than the placebo arm to get a recovery. Now, everybody knows the hazard ratio is an intensity rate. It's not really a risk or something like a probability concept, right? So you cannot say this is a 32% or more likely to recover. So basically, this is a wrong way to interpret. So this is actually very interesting. Because you have a competing risk, you're basically handling two subdistribution function, right? Basically, you are not going to observe this, the blue curve towards the end, it's like reach to 1, because you have those people died. You're never going to observe those people, even you extended your follow-up for 1 year, right? So when you have a subdistribution function, you define the hazard ratio. It's very hard to explain. But my student, the older student, Jason Fine, actually wrote a very nice paper with my friend Bob Gray at Dana-Farber. That's one of his thesis paper, actually. See, they actually pretended to just artificially define the hazard ratio. That's why people use it quite a bit for this hazard ratio for subdistribution function, even though we have a hard time to understand. Even the Jason Fine's paper, they admitted that it's very difficult to explain. But in any event, basically, the NIH study, they actually just use this number with a p-value significant, right? They claim that's it, right? They actually don't have much to say more than that statistically, okay? So it's very difficult to interpret. Now, hazard ratio under competing risk, as I said, is very difficult, right? So what is the alternative way to summarize? Let's go back to the curve now. If you say, hey, listen, how did you summarize each curve, right? I said, well, you told me the higher the curve, the better, right? Then I said, do you think the area under the curve is a good way to summarize the curve then? I said, well, yeah, I mean, the higher means the area under the curve is larger, right? So that means the larger the curve is better, right? I said, well, can we use this area under the curve, incidence curve, as a summary? I said, well, mathematically it's good, but you have to tell me what does that mean, right? So it turns out this is fascinating. We do have an interesting interpretation for this one. Let me show you. So here it is. You have this placebo arm, right? Incidence rate. You have this area under the curve up to 28 days. And we calculate the area under the curve for 11.9 days. And for the treated arm is 14.1 days. I said, well, how do you explain to people? What do you mean by this? This is actually the average, the number of days across all the patients. They actually happily enjoying called a post-recovery time. That's exactly the one I like to emphasize in the plot. Sorry, look this on, right, the right-hand side. If you forget about censoring observation, right, you're just adding those right-hand side number and divided by number of patients. So that means the average time that people actually happily say, oh, I'm out of the crisis, right? I mean, I enjoying the number of days. So that's the area under the curve we're talking about. So then the question is 11 days, 11.9, the shorter, of course, the worse, right? So the difference between the two arms is, is 2.2 days, right? The difference. The confidence interval is 0.89 to 3.52. See the p-value, highly, highly significant, right? But you can argue, say 2.2 is clinical meaningful or not. I believe in this case is yes. But people also use the median recovery time, right? So how do you do the median recovery time? So you go back to this curve. You say, what is the median? The median is 0.5, right? You're reading those guys, right? So you have a 12 days against 16 days between the two arms. Now be careful. If your mortality rate is very high, sometimes you cannot even got 50%, right? So basically, the median is not a very good summary. It is a local measurement. We don't even worry about the long-term, the event time or short-term, right? It's basically just 50%. But in the paper, ACTT1 used this median recovery time as a clinical summary, but they never did any analysis at all. How do we actually compare the two medians? Okay. So the lesson we learned from ACTT1, competing risk issues, right? Cumulative incidence curve, and the paper, NIH people didn't use this competing risk issue, but I believe this is not very serious. I think one thing we could modify in the future, don't use a hazard ratio for this competing risk setting up, right? We really need a clinically interpretable estimate, right? So I actually, I'm getting involved in another COVID-19 trial in Europe, and we actually use those average number of days people happily enjoying after recovery as endpoint. Now, next one, I better speed it up, Sammy. This is actually steroid, right? This is done by UK people called Recovery Group. This is also published in New England Journal of Medicine. You notice all the hot stuff collected by New England Journal of Medicine, right? My friends, all the associate editors, boy, I tell you, they enjoying all the publicity. I think I was told that if you publish one paper in New England Journal of Medicine, you probably get a promotion pretty soon. So that's what happened to this New England Journal of Medicine. But in any event, but we are actually lucky. In the ACTT1, we actually wrote a letter to the editor, New England Journal of Medicine, saying you shouldn't using hazard ratio. Actually, we are so lucky. They accept the publication in July last year. And amazingly, I got lots of emails and asking me about this concept. Now, anyway, so this is study endpoints of survival. Well, either time to survival or time at by day 21. Okay, so this is a interesting trial. They actually in the beginning they didn't stratify the patient based on the baseline severity of disease. So after in the middle of the trial, they find out, oh, wait a minute, doesn't look good. So they actually post-stratified into three groups. The first group is less severe. The second is middle. The third one is severe, right? The severe one actually, it turned out is very interesting. But the question is when you're dealing with the time to death, right? In the short-term trial, should I worry about the survival time? How much shorter with my treatment compared with a placebo, or really worry about like a 28-day survival rate, right? Interesting enough, they actually use a hazard ratio in the paper, right? So the people actually have this habit, right? Every time you involving time to event, everybody jumping on the wagon and say, let's use a hazard ratio, okay? So, so let me show you the hazard ratio first, then I go back. The hazard ratio is a 0.87. And is significant. Okay, I'll adjust it. They did adjust it later. So I say, well, gee, hazard ratio is not a really good. What is alternative now, right? Even in a chronic disease, we find out hazard ratio, the proportion hazard model may not be okay. So then we actually, our group study very hard. We always ask Lu Tian's advice to say, how do we do this? So now we actually use the area under the curve of the Kaplan-Meier curve to summarize. We call this a restricted mean survival time for 28 days, right? So in this case, again, the curve the higher, the better, right? That's the Kaplan-Meier time to death. So that's 23.8 days against 23.1 days. Okay, this is interesting. You only gain 0.7 days from the treatment, right? Highly, highly significant. So you guys are saying, well, do you really think a 0.7 days advantage from the treatment is really clinical relevant? I said, not really. I said, what is really relevant? It's the 28 days event rate here, survival rate, right? 77 against 74. okay, which is marginally significant. So I think the problem for clinical care with short-term survival analysis, we shouldn't even bother to using hazard ratio or using the time to death, those endpoints. That basically, we only worry about the 28 days survival rate, okay? So let me give you another example. This is an ongoing study called ACTT3 now. ACTT3 evaluate a combination now. See, everybody now is jumping on the wagon using combination against the remdesivir alone, for example. Again, they use a time to recovery, right, from day 1 to day 29. It's the same as ACTT1, okay? And we don't know how they designed the trial. They use a hazard ratio to design it. They didn't say much about it. Those PIs, they don't want to share the information with us, okay? Now, interesting enough, they have a 33 secondary endpoint, right, in this ACTT3. Now, if you look at this, the secondary endpoint, look at this, the endpoint, duration of hospitalization, duration of invasive mechanical ventilation, duration of new oxygen usage. So it uses all the durations. Now, here is a bigger problem. Suppose your treatment has a survival benefit, right? People tend to survive longer. If you use the duration of hospitalization, what happens? You actually, your patient in the treating arm is penalized because they live longer, right? So they stay in the hospital longer. So this is a really bad endpoint if you expect to have some kind of mortality benefit from your treatment, right? Instead, you don't use the duration of hospitalization. Using hospital alive free, right? Free survival. That means how many days after you checking out from hospital alive? Exactly the one we're talking about, right? The lifespan, people enjoying recovery. But right now, just the time spent, people enjoying after hospital discharge, right? So then in that case, you only worry about those survivors. So clinical speaking, that makes sense because who cares? Those guys, if the guy died, I say, what is your duration of hospitalization? I say, I don't know. The guys died 10 days after. I said, well, is that 10 days? He said, no, I don't know. I think it should be 29 days or so, right? I mean, it's not well defined. So this is simple things we needed to worry about. So sometimes people take it for granted. But if you really think about it, what is a really clinical meaningful way to do it? Okay, I'm not going to go through this one with you. Let me just lesson we learned from duration of event endpoint. We need to reduce the so-called potential survival bias, right? Consider event-free, like hospital-free survival, right? Or ICU-free survival. Now, funny things, after so many trials were done in the States by Gilead and other people, and this is WHO had this big trial, right? Solidarity trial published October 15, interim analysis, right? They found that those guys, 4 guys, they are useless. They claim it's no effect at all, right? And even you call it hospitalization stay and etc. So this is really interesting. We have no idea what happened, right? Maybe the population baseline, they are quite different. So WHO really badmouths our Gilead friend, right? Say, remdesivir is not very useful, right? So that's what happened. You have this is a large trial. Let's conclude inpatient analysis. That's I think we're running late. We need a model-free and a clinical interpretable summary for the treatment difference, right? For example, using the hazard ratio is not very good because basically you cannot even tell the patient, say, hey, listen, your hazard ratio is 1.32. I said, well, sir, what do you mean by 1.32? The clinical people also get confused. Oh, they said, well, you reduce to 32% risk if you are dealing with a bad event. Then if a good event, you increase the 32% chance getting recovery. I said, well, gee, doctor, what is my recovery hazard or rate? I said, well, I don't know. The statistician didn't give it to me. So basically, a ratio by itself, it's very hard to explain to clinical people or even patients, right? And you have to borrow some kind of a median event time to helping us. Unfortunately, median itself has problem, right? So this poor guy cannot help a hazard ratio too much. So why don't we use this area under the curve, which is a single summary, clinical meaningful, to summarize the time to recovery, but you only worry about what happened to the guy actually afterwards, right, after recovery. So for short-term clinical care study, like the steroid, I don't think the time to event analysis is interesting. So the funny thing is that people use hazard ratio to provide a p-value, to provide the so-called statistical evidence. And again, it's very hard to tell people my treatment only prolong your life for 0.7 days on average for 28 days follow-up. I said, who cares, right? For non-fatal events like a recovery, hospitalization stuff, that's right. We actually helping the hospital, the burden actually reduced, right? Now, the last one is very important, not only for COVID-19. If you have data for each patient, right, for 28 days, and can we actually use multiple outcomes, harm and efficacy, maybe toxicity and et cetera, for patient level, can you come back or say when this guy discharged from the hospital, how much oxygen this guy usage, right, during the hospital? And can we use a multiple outcome, come back with a interesting clinical meaningful endpoint, right? Instead of we do the marginal each endpoint, we give you a difference delta, right? And you have so many difference, and you have a discharge, you have ICU, you have survival, everybody get a difference delta. Then towards the end, you don't know how to connect those deltas, right? So this last one is, I think, is really fascinating. Beyond the COVID-19, apply to any trial. Unfortunately, clinical people, they cannot come with the same sort of consensus, right? And that's why we need all the second opinion, third opinion. I think the next time we should encourage people to have a consultation from a statistician, also second, third, fourth one, right? So we increase our income in some way.
Oh my gosh,
so,
that's a good advocacy for us.
Yeah. Let me, Sammy, sorry. Let me just spend 1 minute talking about how to handle outpatient clinical studies, right?
Yes.
This trial actually just finished a couple, 5 days ago, right? By my friend in Montreal. You know, this PI is a cardiologist, right? They use actually, people knows this one. This is an anti-inflammatory drug, anti-gout, right? And you have the arthritis, right? Very bad arthritis. So this is you tried this 4,000 patients. Look, 4,000 patients. So what is the endpoint now? Endpoint is they actually talking about 28 days people either go to hospital, right? That's an event, or people died, right? So toward the end, you have a 4.7% for treated arm, have 5.8% for the control arm, right? So the difference, you look at this, alterations of 0.79. Look at this p-value. My friend almost killed himself. He said 0.08, right? My point is say this is endpoint selection is very hard because when you have outpatient, first think about it, right? If I don't feel well, right, I probably have a few days after I contract the infection, right, COVID-19, but probably didn't have any symptom yet. So I don't know how long the hiding in my body. Then I suddenly start coughing, I start runny nose, and this stuff, right? I said, well, my wife would say, come on, you're a big man, you can tolerate a little bit. This is a little thing. So I'm just suffering a couple of days. I said, well, gee, Maria, listen, I don't feel good. So I call my PCP, say, hey, sorry, do you think I should have a test of COVID-19? The woman said, well, okay, let me arrange you. I wait a couple of days, getting call. I say, okay, go to hospital, have a COVID-19. I wait a couple of days, getting a result back. I said, oh, yeah, you have a COVID-19 now. What are you going to do? I said, well, there's a trial going on. Let me randomize you. Now, after that, it's already 1 week, right, after, okay? So even in the future, you have this prescribed drug available. People actually cannot be treated very early. So ideally, if you really want to treat outpatient, you need to make this so convenient, this application, right, implementation of the drug. Second, you almost need like over-the-counter medicine, right? So you don't feel good, you say, forget it. I don't even know there's a COVID-19 or not. If I spend $20, I take a couple of tablets, I can reduce my the series of my disease, right? So you see, this is a very interesting now. How do you define the endpoints for this one? In this case, they define very hard endpoint, right? Either people died or something else. You notice that this is trial, only 5 people died in the treatment arm on the bottom, the bullet. 9 patients died in the treatment control arm. And they claim a 44% reduction of mortality rate. You know, they hiding those numbers in the press release. And then we read the paper with, oh my goodness, only 5 and 9? You know, this is the problem, right? You choose such a hard endpoint, you end up clinically, you say, do you really think it's worthwhile? I checked online. It costs you about 10 tablets, about $150, right? So anyway. So this is one ongoing trial. This is also by, I think it's supported by AbbVie and the Vanderbilt University, non-hospitalized outpatient, right, in COVID-19. So this is a combination of the 2 tablets now. And they use this WHO, but they modified it a little bit, right, because it's outpatient, right? So you notice a 1 to 7. I believe they designed the trial using proportional odds model. I would say good luck, right? Towards the end, you probably end up very few on the top, right? Because basically they're not going to die, you know, those outpatient patients, okay? So Sammy, let me stop here, okay? Thank you very much.
Thank you, LJ. And I think it's so important for people, because I received messages even from clinicians who attended this meeting, and some of the BD department attended this meeting. They sent me message about, oh, now we know how important the endpoints are for those different trials. So I'm so glad you go over very clearly on each of those and to understand how important statistics job, design and the endpoint selections. We do have quite some topics or questions listed. Are you able to open the chat box? I know we're all
打过去才行。你挂下来吧，我把那个 end 我再给你打过去。Hello.
I can hear you.
Yeah, sorry. I just muted all. I hear some noises. So LJ, there's a bunch of questions in the chat box. Maybe you can like go through them, like some of the ones that you think they're important.
Hold on. Let me see. I cannot see my on the bottom now. Can you just read it for me? I cannot get on somehow.
Yeah, Sammy, go ahead.
Oh, okay. So I'll just select some since time is limited. So one audience asked, what's your thought about in event-free survival analysis, death and census at day 28 are both assigned 0 days?
Oh, that's a very good question. You know, this is actually people ask the same questions a few times. If you penalize a person, we are still in bad shape on 28 days compare the guy is dead, right? We're both using 0 days. I don't know if you want to use -2 days for death. I don't know. I mean, that's an interesting question. I don't know how to answer this statistically, but some clinical folks probably can put a different weighting, right? But I truly believe if you're extending your 28 days to 60 days, then you probably sorted out this problem, right? If a patient cannot check out of 60 days, I think, well, probably really, really bad shape, right? So you don't have this issue anymore. The problem right now is 28 days. You still have a few patients still in the hospital, right? Still alive, right? So I say, well, you know, I'm sorry you didn't enjoy any days after post recovery during this follow-up time. The poor guy said, oh, please give me a few days. I probably get over with it, right? I said, okay, I'll give you 60 days, right? In that case, this guy probably happily out, right? So I don't know how to answer the question. I think it's a very good question.
There are more, so let's go to another one. Can you comment on the China trial, which actually the first one on the Gilead drug? The results also showed the drug was not superior over the placebo. The trial result was published at Lancet.
Yeah, I know that trial. I think there's people criticize the sample size, I believe, right? That's the case, right? You know, interesting enough, I don't know this particular, I don't remember with so many trials, but in hospital trials, the majority endpoint, they all defined, except for this proportional odds model, right? They actually use recovery or improvement as a definition, or hospital discharge from hospitalization. So I think that trial also using the same endpoint. My understanding, they didn't get a significant result. That if I were talking about the same trial, So yeah, you are absolutely right. We saw that paper very early on, before even Gilead published or NIH published their results.
Yeah. Another one actually you really should answer is, do you know existing packages to do the AUC under the KM care curve? I think you have a package you publish or your student published, right?
Yeah. It's in the paper. I don't know, Sammy, and they kindly forwarded the 3 papers in the paper in Annal of Internal Medicine. There is a quoting say, where is the software you can use? You can click that one. It's very friendly. We actually use that software, design a trial using the time span after recovery as endpoint for Spanish trial right now. And you know, it's very interesting because you can specify as a 4 days advantage, right? You can calculate your number of people needed to achieve the 80% power, for example. So it is available. If you guys having trouble, I can send it to Sammy. This is a link again. Okay.
Yeah. Can you send me the link and we'll try to share in the membership?
Absolutely.
I don't think we have enough time today to go over all the questions. If anyone can follow up with me, I can share the question to LJ as needed, or LJ, you can follow up directly with LJ's email. But LJ, you can see how popular your presentation is. And we kind of went out of time and there's actually some clinician question. I think it's very interesting, but because it's too long and we're out of time.
Yeah, Sammy, can you do me a favor? Can you collectively just collect those questions? Or we can post it in your website, right, after my answer. Do you want to do that?
Yeah, I can do that. We will have volunteer help me to do that for sure.
Okay.
Thank you.
I jump in regarding the China data. I just want to add some information about that.
Yeah,
Is that okay?
Go ahead. Yes,
okay,
of course.
Yeah, the interesting things, LJ, nice to meet you again. That study is early terminated because of outpatient, because the China in Wuhan is totally controlled. And I was involved in that study because we supported the DMC. When we recommended the Chinese government to do continue that study outside, they reject the idea.
Okay. I see.
Okay.
I see.
Now, the conclusion they published in the National, the result is that remdesivir did not do good compared to placebo. There's several reasons. One is that they are based on WHO's earlier definition, they call blueprint guideline, used the 2-scale improvement. Okay, China study used the 2-scale. So most of the patients get into the score 3, you got to be either get out or either. I mean, there's a 2-scale, as you know, it's very difficult to improve. And the US study actually the 1-scale improvement. And also, we also re-analysis data used the method you just mentioned, and also published by I think the DIA Journal of the Open Clinical Access Clinical, which show China data actually is consistent with the US result. So
that's good. Okay.
Remdesivir actually is showing a very positive result significantly compared to placebo. I just want to clarify that.
Oh, wonderful. Well, let me just make a very quick comment. People use the idea with those 7 scale. You say, well, if I have a 2 steps improvement, I call this improvement, right? So people criticize that definition because you can say, I'm in really bad shape, right? I got 2 improvement, that's dramatic improvement. But if you're on the bottom, is you are okay start with, right? Your 2 step is not that interesting. But anyway, any summary has a problem, right? It's not a perfect, right? But I think if you use a different endpoint, we also need some kind of clinical interpretation. The problem we're having in the past, I can speak as a statistician. I'm working on this field for 45 years, even more than that. In the past, we emphasized so much of hypothesis testing, right? So we come back with all kinds of really, actually it's a very unique, people usually don't even understand it. It's like a hazard ratio. People usually don't understand what we're talking about. Odds ratio, common odds ratio, people don't really understand it. But we create those animals in some way. We actually convince people you can get a more powerful test. See, deep in our heart, we really worry about the p-value, right? But nowadays, people say, give me a break. I need more than p-value now, right? So the first hurdle is the p-value. Second one, after you pass the hurdle, you're saying, well, tell me how big the size is. You're talking about improvement, right? That makes sense. Otherwise, how much I pay you, how much I should have recommended this new therapy for you, right? So I think the estimate idea is a very good one. But on the other hand, we actually have to be careful how to define estimate. If you artificially define estimate like a proportional odds model, we still don't know how to explain to people, right? So ICH guideline didn't say this. So we wrote a paper recently. We say, hey, listen, how do I define the estimate? You should have this quantitative, the summary is model free. Otherwise, we're in trouble down the road, right? If your model doesn't feed the data well. So that's basically I want to really share with you. I finally realized in the past 10 years, I finally saying, listen, guys, make life is simpler. If you can make the results of your trial is transparent and interpretable, right, and efficiently enough, that's good enough. And I don't want to badmouth the parametric one. Parametric is good when we do prediction, but when you evaluate a treatment difference, it's not a very good tool. Okay, let me stop here, Sammy.
Yeah, no worries. Actually, I was going to comment the estimate. I mean, the International Estimate Working Group from EU and the US. Definitely, it's a great framework. I totally agree. How we define estimate, it's also interesting topic in order to clarify. Otherwise, the same old problem is still existing over there. But right. in the interest of time, I do want to thank LJ for the great presentation. It really helped the statistician actually explaining how important endpoint is, and beyond that, even all the related thoughts needed to be part of the clinical trials for this topic. So we are having a volunteer in our team already copy all your all the questions from the audiences, and I'll share with you in the email. And when you are able to respond back to us, we'll share with the members in the board in our website. So thank you, LJ, so much for attending.
Sure.
Thank everyone joining us today.
And thank you very much, guys. See you later.
Thank you.
Thank you,
Thank
LJ.
you.
Thank you. Bye-bye.
Bye, guys.
Bye. Have a good one.
Thank you.